Epithelial tissue. Now this video is going to focus on epithelial tissue, what the naming system is, and some of the structural basics of what it actually is in general. The specific tissues we'll get to in a little bit. But first of all, the idea of epithelial tissue, covering and lining. That is the main way you're going to find epithelial tissue in the body. Covering the body, your skin, or lining the inside cavities, lining the inside the GI tract, lining the inside of the lungs, lining the inside of the heart, lining the inside your blood vessels. You get the point, it lines the inside or covers. But you'll also find epithelial tissue as glandular tissue. It makes up various different glands in your body. So functions. Now, these functions are across the entire category of epithelial tissue. So not just one particular one. So there isn't a single tissue that has every one of these functions. This is category-wide. The idea of forming boundaries. Sure, your skin forms the boundary to your body. Protection. Your skin protects. It prevents things from entering into your body. Absorption. If you think about absorption, the cells that line the intestines, they're absorbing your nutrients. Filtration. Look into your kidneys for that one. You're going to have epithelial cells in your kidneys that help to filter out the blood. Excretion, secretion. Take your pick. You can go into the kidneys, you can go into the intestines. And sensory reception. Now, there are different adapters or structures that are part of your skin, for example. They'll help detect pressure on your skin. They can help with sensory reception. All right, let's look at some special characteristics. Now, these special characteristics are again for the whole category of epithelial tissues. But these characteristics hold true for pretty much every one of the epithelial tissues. The idea of cellularity. In cellularity, it's talking about how all the cells are very close together. There's not really a lot of space between the cells. That means you don't typically have a lot of extracellular material. It's just one cell, two cells, three cells. They're all very close, very tight. So it's almost all pure cells without things in between. Special contacts. Now, throughout epithelial tissue, this won't happen with every tissue necessarily, but most of them have a special contact, something that helps to hold things together. First example is tight junctions. Now, if you notice in this diagram, it's all the blue kind of dots going through. It's almost like someone took and stitched the two different cells together, taking those cell membranes and sandwich them very close, very tight. This helps to prevent anything from passing between the cells, which means anything going through this layer has to go through the actual plasma membrane. It can't just sneak between the two cells. Tight junctions. Gap junctions. Now in this case, again, it's two cells together, but you're going to notice these are actually pores. They're channels. They allow ions and small particles to travel from cell to cell. This can aid in cell-to-cell communication, for example. So we have these gap junctions, allowing ions or particles to travel from cell to cell through these specialized channels. And then anchoring junctions. Now there are different ones here, from desmosomes to hemidesmosomes to adherens. But what they're all doing is they're all taking two cells and helping to hold them or anchor them together. They're not quite the same as your tight junctions because there is still a little bit of space there. But it's helping to hold the cells to each other. Either keep them cell to cell for the epithelial tissue or anchor them down to the basement membrane below. But regardless, you're looking at these proteins that are anchoring to hold them in place and keep them together. These are all creating structural elements inside of these tissues. So some specialized structures there, helping to hold things together. Next up is polarity. You may have heard the term polar opposites. Think about a battery has a positive and negative, or the North Pole and the South Pole. The idea is polarity has two distinct sides to it. In the case of epithelial tissue, we're going to have two distinct sides to the tissue. You're going to have both the apical and the basal side. The apical side is going to be the exposed side. In the case of your skin, you're touching the apical surface. In the case of your stomach, for example, the inside lining is epithelial tissue. The part that the food comes into contact with, so if this is your stomach, this inside part here, this would be where the food is. That's where you're coming in contact. That's the apical surface. The basal surface is what's always in contact with other tissues. So the basal surface is never exposed. Now this apical basal orientation allows us to also have the basal side being a basement membrane. You can see here we have the basal lamina and then the reticular lamina. The basal lamina and the reticular lamina come together and make what's called the basement membrane. It's the junction between the epithelial tissue and the supporting connective tissue below it. You can see you have down here glycoproteins and collagen. Well, those are going to be structural units found within the basal lamina, helping to give it support and structure. The reticular lamina, collagen found from connective tissue. We'll talk a lot more about collagen coming up in the connective tissue section, but collagen is going to be a fiber that provides strength, high tensile strength. So if we're looking at the basement membrane, so we're looking right here, you can see the basal as well as reticular lamina. It's those two areas that will make up the basement membrane between the epithelium, which is up top here, epithelium, and then connective tissue below it. What we're doing is creating this network, this structure to help hold the two different types of tissues together. And it's going to be important to keep this in mind as you go for more of these special features. But basement membrane, both the basal lamina and reticular lamina together making up your basement membrane. I already mentioned supported by connective tissue. Well, sure, it's supported by connective tissue. We just mentioned that basement membrane helps to anchor epithelial and connect it together. The question is, why does it have to be supported? Your epithelial tissue is avascular. So if you think of the term avascular, A means without, vascular refers to blood. So avascular is without blood. Well, hold on. Blood brings nutrients. Blood brings you gases like oxygen. It's kind of important. The way this works is your epithelial tissue is avascular, but it's always supported by connective tissue. Connective tissue is vascular. That means the blood that's in the connective tissue will have the nutrients and the gases diffusing through the basement membrane up into the epithelial tissue. The nutrients and oxygen needs of the epithelial tissue are provided by the supporting connective tissue. And that's why all epithelial tissue has to be supported by connective tissue. But while epithelial tissue is avascular, it is innervated. Innervated means it has a nerve supply. So you kind of think of this one. Maybe you've all of a sudden fallen down and skinned your hand. But as the skin kind of peels back, does it always bleed? Not always. The reason being is if it peels back but doesn't bleed, you've only damaged the epithelial tissue. You haven't cut deep enough to go to the connective. So there's no blood. But does it still hurt? Oh yeah, it still hurts. That's because it's innervated. So the epithelial tissue, avascular, no blood, but it is innervated, has nerve supply. And your epithelial tissue is regenerative. It means it's highly mitotic. Mitosis, one cell divides to two. Remember, mitosis is a perfect copy. So that first cell has a perfect copy to the next two. What's doing here is it constantly creates new layers of epithelial tissue, new ways of layering and protection. So how do we classify? How do we name our epithelial tissue? It's named two different ways, by one, layer, two, shape. Let's start with the shapes. Squamous. Squamous are flat cells. They're like pancakes. They're very thin. So squamous cells, very thin cells. Cuboidal. Cuboidal are box-like. They're roughly a cube. Now, will you ever have a perfect 90-degree angle? Probably not. This is nature, this is human body. Their curves will be more rounded, the corners. But they'll be more box-like than anything else. So the idea of cuboidal. And then we have columnar. Now, columnar is going to be more tall like a column. Think more like a tall rectangle. Again, not perfect corners, but you're looking at the idea there. And these tissues, whether it's squamous or cuboidal or columnar, they can be that single layer or they can be multiple layers. So you have the idea of simple, one flat layer, or stratified, two or more layers. So simple versus stratified. And then we have that last little bottom corner there called pseudo-stratified. Pseudo, the prefix, means false. Pseudo-stratified, false layers. That means in this case, that pseudo-stratified, every single cell is touching the basal surface. So every cell is touching the basal surface right down here. But not every cell reaches the apical surface up here. So in the case of pseudo-stratified, it's only one layer because every cell touches that basal membrane. But not every cell gets the apical, so it looks like you have different heights there. So simple, stratified, pseudo-stratified are the layering, which means the two naming system. This would be simple squamous epithelium. Simple, one layer. Squamous, flat cells. So simple squamous, one layer flat cells. All this naming is going to help to identify the tissues based on what you're looking at. Then we have this thing called transitional. Transitional, if you notice, there's nothing else popping up but that word. Because transitional, that tissue can have cell shapes of squamous, of cuboidal, and of the actual tall rectangular. So looking at columnar. The idea here is, and we'll get more into this, transitional is found in the urinary bladder. It's going to change shape, so the cells themselves change as well. So that transitional doesn't have any dedicated shape. It could be any of the three shapes, squamous, cuboidal, or columnar. That's the idea of shapes and layering, but just to reiterate the layering and show kind of an own slide here. You have simple, again, one flat layer, that's it, single layer. You have stratified. Stratified is going to be two or more layers. So it could be 2, could be 10, could be 5, whatever. And then you have the pseudo-stratified. Pseudo-stratified is one layer that looks like many. Because again, one layer, everything's touching the basal surface down here. Not everything reaches the apical surface up there. So we're looking at the two different layering looks like. It looks like more, but it's only one. Pseudo-stratified. Some specialized structures you'll also find in epithelial tissue. Cilia. Now, cilia, that's this hair-like projections here, those cilia are going to protrude from the actual cell. So they're protruding from the cell. You're going to find ciliated or non-ciliated only with columnar epithelium. So if you see cilia, you know it has to be columnar. This particular example, this is going to be some pseudo-stratified. So we have, looks like more than one layer, but it's only one. But they are columnar. So pseudostratified columnar with cilia. Microvilli. Now this example is coming from your intestines. The microvilli are going to be small finger-like protrusions coming up from a cell. So these are the individual microvilli right there on one single cell. To put that in perspective, each one of these cells right here has all those microvilli on it. And they're making up only one villi of the entire structure of your intestines. So each one of these parts, this tiny little protrusion, each one of those little finger-like projections is a single villi over here, which then you have the microvilli on each one of those cells. So there's a lot of different parts and pieces all coming together to make up your intestines. But microvilli, small finger-like protrusions that increase surface area. It's all meant to increase surface area. Then we have goblet cells. Goblet cells, those again are only found in columnar epithelium. So goblet cells. In this case, you can see the goblet cell is located right there. That's going to be a goblet cell. Goblet cells are going to produce mucin, basically mucus, think of it, mucin, for secretion onto the surface. So our goblet cells. So that's the basic overview of epithelial tissue. Next, we move into what the specific types are, where to find them, how to identify them, and what their functions are.